right, let's get right into this. Welcome to your high-yield, exam-oriented explainer on microscopy. If you're an MBBS student gearing up for exams, you are in the exact right place today. We're going to completely transform how you look at the microscopic world. I've designed this session specifically to help you crush those microbiology MCQs and absolutely ace your short notes. We'll be relying heavily on mnemonics, tables, and crystal-clear breakdowns to make sure these concepts really stick with you. Here's your mental roadmap for today's explainer, following the exact structure you need for your exams. We're going to start with a quick introduction, then move through the bright field light microscope, phase contrast, fluorescence, and we'll wrap up with the highly detailed electron microscope. Starting things off with Section 1, Introduction to Microscopy. Now, before we talk about the tools themselves, we kind of need to know what we're actually trying to look at, right? The crucial thing to grasp here is the absolute scale of these pathogens. You'll want to remember this fundamental scale for your MCQs. Viruses are literally tiny, measured in nanometers, specifically 20 to 300 nanometers. You absolutely have to use an electron microscope to see them. Meanwhile, bacteria and fungi are a bit bulkier, measured in micrometers. Bacteria typically sit right around 0.2 to 1.5 micrometers in diameter. Because of that size, you can comfortably visualize them under your standard light microscope. Okay, want to guarantee full marks on a short note about microscope properties? Just use the mnemonic RCM. R stands for resolution, which is the ability to produce separate images of closely placed objects. C is for contrast, which we usually improve by staining our specimens. And M is for magnification. That's just the simple math of multiplying your 10x ocular lens by whatever objective lens you happen to be using, like 100x for oil immersion. So remember, RCM. Let's talk a little bit more about resolution, because trust me, examiners love to test this. Keep this specific high-yield number in mind, 0.2. The unaided human eye can only resolve objects down to 0.2 millimeters. But a standard light microscope, it actually resolves all the way down to 0.2 micrometers. That is literally 1,000 times better. Moving right along to Section 2, the light microscope. If you get a question on the bright field microscope, you need to be ready to reproduce its core components. The easiest way is to mentally group them into 3 distinct parts. First, you've got the mechanical parts, the base, the C-shaped arm, and the stage. Second are the magnifying parts, the ocular lens up top and the revolving objective lenses. And third, the illuminating parts down at the bottom, the light source, the condenser to focus the light, and the iris diaphragm to actually control it. Now, check out this brilliant little modification. Notice how the standard bright field pathway compares to the dark field pathway? See how the dark field condenser uses a central opaque disc? Well, that opaque area totally blocks direct light from hitting the lens. This means only the oblique, unreflected light that actually bounces off the specimen makes it into the objective lens. It's a really clever trick. So why do we even care about that? Because dark field microscopy is an absolute favorite for exam topics. The result of blocking that light is a bright object shining against a pitch black background. Your high-yield targets here are basically twofold. 1, examining living unstained cells. And 2, definitely highlight this in your notes, visualizing incredibly thin bacteria like spirochetes. They're just simply too thin to be seen with a standard bright field light microscope. All right, Section 3, phase contrast microscope. We all know staining kills cells, right? So how exactly do we get good contrast if we want to study living microbes without destroying them? The physics here are actually super elegant. When light passes through a specimen, let's say a bacterium, some rays are deviated and slowed down because of the cell's density. Other rays just pass right by, totally undeviated. Inside the microscope's objective, there's a special phase plate. The undeviated light passes through a phase ring and gets advanced by exactly 1/4 of a wavelength. So when it finally catches up and meets the deviated light, boom, they're exactly a half wavelength out of phase. They end up canceling each other out, which creates this incredible contrast in a living, transparent cell. Okay, so how are they going to test you on this? If a multiple choice question asks you how to best observe microbial motility, your answer is phase contrast. If they ask how to detect internal bacterial structures, things like endospores or inclusion bodies in living cells, again, the answer is pretty much always phase contrast. It's the go-to tool for converting slight refractive index differences into stark visual differences. On to Section 4, Fluorescence microscope. Now, this technique uses invisible, short-wavelength ultraviolet rays to generate a visible, glowing image. Here's how it works. An excitation filter lets in only those short UV rays. These rays hit your specimen, which you've already coated in a fluorescent dye. The dye gets super excited by the UV energy and immediately emits light of a longer wavelength, which conveniently falls right in the visible spectrum. That emitted light travels up through a barrier filter, which keeps your eyes safe from any leftover UV rays, and the result is stunning, glowing images like the bright green tubercle bacilli. The absolutely crucial point here for exams is matching the specific dye to the specific bug. You definitely want to memorize this table because examiners love, and I mean love, to test your knowledge of these fluorescent dyes. If they ask about autofluorescence without using a dye, you should immediately think cyclospora. If they mention acridine orange, that's your specific trigger for plasmodium and filarial nematodes using the quantitative Buffy-Coat method. And of course, the classic. Auramine phenyl is the absolute go-to for identifying tubercle bacilli. Make sure you lock those pairings in. Finally, Section 5. The Electron Microscope. We are totally leaving the micrometer world behind now. We're diving deep into the nanometer scale with the ultimate tool for viral detection. It's crazy how completely different an EM is from a standard light microscope. Look at the towering transmission electron microscope, or TEM, and its internal pathway. Instead of a basic light bulb, it uses an actual electron gun. Instead of glass lenses, it uses powerful electromagnets to focus the beam. And all of this has to happen inside a high vacuum. Because electrons have incredibly short wavelengths, the TEM achieves an astonishing resolution of 0.5 nanometers. This is exactly what you need to detect viruses directly from clinical specimens, like identifying rotavirus in a stool sample. But here's the catch. You can't just slap a raw specimen under an electron beam. It would literally be obliterated. Specimens have to be carefully prepared to survive both the high vacuum and the intense electron bombardment. So definitely lock the FDES mnemonic into your memory for any short notes on specimen prep. F is for fixation, using glutaraldehyde. D is for dehydration, using ethanol. E is for embedding it into a really hard plastic polymer block. And S is for slicing it with an ultramicrotome knife down to an unbelievably thin 20 to 100 nanometers. So that's FDES. Now, if you get a short note asking you to compare light and electron microscopes, reproducing these high-yield points will pretty much guarantee you top marks. It really brings the whole explainer together. Light microscopes use visible light, glass lenses, and they max out at around 1500x magnification with a 0.2 micrometer resolution. Electron microscopes, on the other hand, use an electron beam, magnetic lenses operating in a high vacuum, and they can magnify well over 100,000x with a stunning 0.5 nanometer resolution. Oh, and definitely notice the mount difference. Glass slides for light, but copper metal grids for electron. Okay, before we wrap this all up, let's do a quick knowledge check with a really classic direct exam question. Which microscope makes the object appear bright against a dark background? Based on everything we just covered, the instrument that uses that opaque disk to produce a bright object on a pitch-black background is, yeah, you guessed it, the dark ground or dark field microscope. Ultimately, mastering these properties, these handy mnemonics, and the core physical principles isn't just about passing an exam. I mean, it will definitely help you do that for sure, but it's really about recognizing how pushing the physical boundaries of what we can see allows us to identify dangerous pathogens, understand intricate cellular structures, and at the end of the day, save human lives. I really want you to think about that next time you're looking down an ocular lens. As our technology keeps pushing resolution closer and closer to the atomic level, what invisible threats do you think we'll discover next? Thanks for learning with me today, and I'll see you in the next explainer.